I'm going to take a deep, lightly
Do you know who that is? Do you remember? That's not Jake
All right. Morning, everybody. I'm not sure if the grades already posted, right? For exam 2. No? I think you guys said it better than the exam 1. Oh, you guys.
I checked this morning anyway.
Oh, average is 42 point something, can't remember, but last exam 1 was 40, right, 40 point something, so I think better. So you guys did great. So today we're going to talk about protein metabolism, protein synthesis, degradation, and then the next 3 sessions, Dr. Kerry Erzman will talk about the amino acid metabolism. Right? Okay, so functions of functional proteins. Proteins usually account for about 70% of our body mass, right? And the main functions of proteins include, first, maintain the body's structure, right? The collagens, protein very important, and this is important function to maintain our body structure. And secondly, protein and also serve a important structure proteins and facilitate mobilities, particularly in the muscle, right? So like actin and myosin are the important structure protein in skeletal muscle, right? The muscle tissues. So that involve the the contraction, muscle contraction. And thirdly, proteins also serve as important transporters. We already know, like hemoglobin, right, is important protein that carries oxygen from the lung, right, and to the other tissues for metabolism, particularly for oxidative metabolism that requires oxygen. And membrane transport system, right, we learned some of the transporters, for example, glucose transporters, right? It's very important for glucose metabolism and maintain blood glucose levels. So protein, serves as transporters, and also we know we have other transporter proteins like amino acid transporters and fatty acid transporters, right? All these transporters are proteins, so protein important. And then protein also involve the metabolism, right? All the enzymes that we learned are proteins. And protein also functioning as the regulatory factors, for example, hormones, right? Peptides, the growth factor, they are all proteins. And lastly, the immune function, right? Protein is very important for immune function, like immunoglobulins and cytokines, these are all proteins, right? And finally, protein can also be used for energy, although it's not the most important function, but protein can be used for ATP production, particularly under the fasting condition, right? We don't have enough glucose or fatty acids as energy, so protein can be used for energy. And here is the overall of the protein and amino acids metabolism. In our body, there is a so-called amino acids pool. So when we eat food and we digest the proteins and absorb the protein in the form of amino acids, right, in the body, so that will contribute to the the body amino acids pool, and amino acids in our body can be metabolized in several pathways, right? In several different ways. One is the protein turnover. So amino acids, once absorbed from the diet, can be used for synthesis of protein, right, and stored in the body or for body need. And during the fasting condition, that protein can also be break down back to amino acids, and either for energy use or further metabolize or synthesize other important molecules, right, in compounds in the body. And the second important pathway is the catabolic pathway, right? As I mentioned, amino acids can be metabolized for energy use. So because amino acids has a nitrogen in addition to carbon, right? So, so for amino acids metabolism, catabolism, first of all, we have to remove the nitrogen, right? So nitrogen metabolism include nitrogen can be metabolized to generate ammonia, and also can be used for synthesis of urea, right? So this is one way that amino acids nitrogen can be metabolized, and the carbon in the amino acids can be metabolized basically for energy use, right? So first is the amino acids can be oxidized to generate acetyl-CoA. Acetyl-CoA is a common, the central molecule, right? So glucose, fatty acids, and amino acids, these three nutrients can all be metabolized to generate acetyl-CoA. So acetyl-CoA eventually enters the TCA cycle for ATP production and complete oxidize to generate CO2 and produce energy, right? So this is also the important one of the important pathways of amino acids catabolism. And the other thing I wanted to remind you that it's a little bit different for amino acid metabolism in the fat condition. Amino acids, like similarly to glucose, if we have a little bit more, if we have more amino acid that we need, the amino acids can also be converted to fat or glycogen and stored in the body, right? So in the fat condition, when we have excessive amino acids, and these amino acids can be metabolized to generate the TCA cycle intermediates. Remember, we talk about the TCA cycles and gluconeogenesis, right? So one of the intermediate oxaloacetate, right, that can be produced from acetyl-CoA, and this oxaloacetate can enter the enter gluconeogenic pathway to generate glucose. But this glucose will not be released into the circulation and for use in other tissues. Instead, the glucose eventually be converted to the glycogen and stored in the body in the fat condition, right? So, and also under the fat condition, the acetyl-CoA that generate in the in the catabolism of amino acids can also be used for fatty acid synthesis and triglyceride synthesis. So, and stored in the body as the energy for use later during the fasting, right? So, so this is the how carbon in the amino acid can be metabolized under the fat condition. But I also wanted to tell you, I mean, although it's not indicated in this slide, under the fasting condition, right, the amino acids usually compete, oxidize for energy, and also amino acid can be used to synthesize glucose, right, gluconeogenesis, and this glucose will not be converted to glycogen, but instead, released into the circulation, right, to raise blood glucose level. So it again, it's a different metabolic fate or pathway under the different physiologic condition, fasting condition, right? And in addition to all this metabolic pathway, that amino acids can also be used to synthesize non-protein derivatives, for example, purine, pyrimidine, neuron and transmitters, and non-peptide hormones. And this can, factors that can, or compounds can also be synthesized from the amino acids, right? Any questions? Okay, so in the general metabolic pathway protein, that we can divide it like two opposite pathway, like protein synthesis and protein degradation. So protein synthesis, let's talk about protein synthesis first. So protein synthesis involved in the transcription and translation, right, process at the molecular level. Transcription, that means the DNA can be transcribed into three types of RNA in the nucleus, and that includes messenger RNA, transfer tRNA, and ribosomal rRNA, right? And then translation stage, the messenger RNA can be translated into protein, right? With the help of the other two types of RNA, tRNA and ribosomal RNA in the cytoplasm. So protein is synthesized in the cytoplasm. Transcription happens or occurs in the nucleus, and that involves the three types of microRNAs, right? Messenger RNA is a genetic coding template. It contains the genetic information. So the amino acids sequence, or the orders of amino acids that is determined by the messenger RNA template, right? So so messenger RNA serve as the genetic coding template, and transfer RNA, tRNA. So tRNA, what tRNA does is to transfer individual amino acids to the messenger RNA template, right, for the translation. And so basically, carry the amino acid to the site of the translation. And ribosomal RNA usually form the ribosomal with ribosomal proteins, and serve as a platform, right, for the translation or for the synthesis of proteins. Usually have two, the ribosomal subunit, and form the whole template, like 40S, 60S ribosomal unit. They bind together, right, and form the the template for protein translation. That's pretty basic information, right? You guys learned from biochemistry, I guess. So, and I think we don't need to include, or remember the very complicated process, but just the overall, protein synthesis can be divided four stages. First is amino acid activation, and initiation, elongation, and termination, right? The fourth stage, I don't know what that is. And the basic components that, the materials that needed for protein translation or synthesis. First of all, basic components like amino acids, right, is serving as blocking molecules, and RNAs, like 3 type of RNAs, right, messenger RNA, transfer RNA, and the ribosome RNA, and also requires energy, right, ATP and GTP is also needed for the translation process. And because it's also involved, the multiple stage, like initiation, elongation, and releasing stage, and all this process requires enzymes and proteins called factors, right, involved to regulate, each process stage. And of course, the enzyme, right, is needed for the process of protein synthesis, and ribosome, I just talk about, it serves as sort of a platform, right, for the synthesis of proteins. Just know that basic or general steps, right? Don't need to remember which factors, enzyme that involve, right, specifically. And for amino acids activation, that is sort of the overall reaction. So you see that amino acids basically need to be attached to the transfer RNA. We know that transfer RNA serve as the carry RNA, right? So carry amino acids from one side to the translation side. So first of all, the specific amino acids that have to be attached to the transfer RNA in order to be carried to the translation side. So this process is called amino acid activation. This process takes 2 ATPs, right? You need energy for doing this, and the enzyme that called amino acyl tRNA synthetase, and this is enzyme to catalyze the amino acid activation. And we have our body has 20 amino acids, right? And only one, the enzyme, the amino acyl tRNA synthetase can activate 20 individual amino acids, right? So one enzyme activates 20 amino acids. And the affinity, this enzyme affinity for amino acids is high. That means that even when we have a lower amino acid concentration in the body, the protein synthesis still can happen, right? Because they have a high affinity. So it's very easy to bind the amino acids and activate amino acids. Yes?
The activation occurs in the cytoplasm or
It's in the cytoplasma, so then can carry, because the translation process occurs in the cytosol, right? The transcription, process occur in the nucleus, right? Okay, so post-translational modifications. So once the protein is synthesized, very primary sequence, right? Very preliminary. So primary sequence of a protein once it's synthesized through the translation process, and this need to be further modified in order to be, active. So for example, enzyme, right, need to be active. So there are actually multiple modification process. And for example, one is you have to remove the N-terminal methionine residues. The N-terminal methionine residue is required for protein translation process, but it's not needed for protein activity, right? So first of all, this N-terminal methionine residue need to be removed, and then the cleavage unnecessary regions. So during the translation process, some additional sequence is synthesized, but this sequence again is not needed for protein function. So this unnecessary sequence is also need to be removed, cleaved, and also some kind of specific modifications, for example, glycosylation, acetylation, and fatty acylation. So, and this means that, for example, the glycosylation, that means the sugar group can be added to a certain amino acid residues of the protein, and this modification is also important in terms of the regulation of protein activity or degradation, something like this, right? And acetylation means that acetyl group can be added to the protein, amino acid residue of the protein. And acetylation is very recently been found, is very important regulatory mechanism. Once the protein is acetylated, and activation can be changing. The activity can be changing, either increase or decrease, depending on the proteins, right? Like a phosphorylation, dephosphorylation. And other one is fatty acylation. Similarly, fatty acyl group can also be added to the the proteins, right? Modified protein. And also the protein, once the primary sequence is synthesized, lots of proteins also need to form secondary, right? And even quadratic structure of a protein. So sulfur bond formation is also important. Can connect the two single the amino acid sequence, right? In order to be form the more complex structure for the activity of protein. And finally, protein also need to be folded, not just linear, but need to be folded. So folding of protein that occurs usually in the ER. Right? So once all this process is complete, then protein can be activated, right? It can have a function. So just you probably wanted to know that what are the components necessary for protein synthesis, like amino acids as a building block molecules, and 3 types of RNAs, and then lots of initiation, elongation, releasing factors, right, and enzymes, kind of needed. But again, you don't need to remember specific, right, names of enzymes. And 4 stages are involved in the protein synthesis, right? Activation of amino acids means that attach amino acid to the transfer RNA for for being transported to the translation site, and initiation, elongation, and termination, right? The 4th stage. And each stage will have a specific regulatory mechanism to control the rate of protein translation. Okay, so that's just for protein synthesis. Now let's talk about protein turnover. So protein, once it's synthesized, they're not like present forever, right? They can be degraded, and new protein need to be synthesized. So protein usually constantly being synthesized and degraded, right? So protein synthesis and degradation, this 2 processes that's together and form so-called protein turnover, right? So the concentration of proteins in cells usually is determined by a balance between the rate of synthesis and degradation, right? For example, if somehow a synthesis process is go more active, then or degradation process somehow decrease, and that will cause the protein concentration will be increased, right, in the body, which is probably not good, right? We want to just right amount of protein and have right, levels of activities. So if protein level is too high in the body, particularly if some like enzymes, right, or some other key regulatory factors, if the concentration too high, then can causing some dysregulation of metabolism, right? So again, the balance is very important, the protein turnover process. And positive balance, protein can result hypertrophic, increase the protein content in the cell, which causes some dysfunction of cells and some problem of disease conditions. And negative protein balance can result atrophic. So loss of protein from the cell, again, is also not the right thing, right? It's also causing some problem in terms of cell function. So now let's talk about protein degradation. So protein degradation, our body has several pathways that can regulate protein degradations. So in general, the three major pathways, right? First is so-called lysosome pathway, and lysosome pathway usually degrades the extracellular and the cell surface proteins, and also the intracellular proteins, right? So lysosomal pathway is very important pathway, and that can degrade, extracellular protein, for example, protein present in the plasma, and the cell surface protein, and also intracellular proteins. Oh, I'm so sorry. And then the second pathway is the proteasomal pathway, right? Proteasomal pathway basically degrades the proteins from the cytoplasma, plasma and nucleus and ER in the cells, right? Intracellular cells usually degraded by the proteasomal pathway, right? And the third pathway is called cytosolic proteinase. So there are also a lot of proteinases that are present in the cytosol that can also regulate the degradation of intracellular proteins, right? Now let's talk about lysosomal protein lysis, right? The pathway, how this pathway is regulating protein degradation. Any questions so far? Okay. Oh yeah, sure.
Like the main pathways,
Uh-huh.
the most important pathway is lysosomal. Oh, never mind, different thing. Oh. So
Right.
Is this the first pathway that you were just talking about? Or?
Yeah. See, we have this. These are the three main pathways, right, for protein degradation.
Okay.
And the next slide, this slide, I'm going to talk about specific, each pathway, right? So this will start to talk about the lysosomal pathway. Yeah, it's very important pathway, right? So the first main pathway is the lysosome pathway, right? So lysosome basically is a membrane enclosed vesicles, right? Have you guys learned, right, about the organelles, right, in the cells of lysosome? Lysosome is the membrane enclosed vesicles that initially formed from the Golgi complex, right? So and inside there are actually as many as 60 different type of digestive enzymes inside lysosome. So lysosome contains more than 60 digestive enzymes, and this digestive enzyme can degrade protein that once is internalized in the lysosome, right? And this pathway depend on the substrates of the degradation. can be further divided into four different categories, right? First is like vesicle fusion, and second is a macro-autophagy, and third is micro-autophagy, and lastly is a direct transport by chevrons, right? So 4 different mechanisms that involve the lysosomal degradation pathway, right? Is that clear? I mean,
Does that mean that
yeah.
Like the breakdown parts just contribute to all these different processes? Or are they, I'm just trying to understand, I don't really get it.
No, I would say that protein can be degraded through a lysosome pathway in 4 different ways,
Oh, okay.
right? Is that clear? Right. So one is like vesicle fusion,
Okay.
macro-autophagy, and micro-autophagy, right?
Okay, got it.
Okay. Okay, so again, the lysosomes deal primarily with the extracellular protein, the degraded extracellular proteins, right? For example, protein that are present in the plasma circulation that can be taken up into the cell by the endocytosis mechanism or internalized right into cells and for degradation in the lysosome. And second type of a protein that can also be degraded by lysosome pathway, and that is cell surface membrane proteins, right? That can be used in the receptor-mediated endocytosis pathway. Do you remember which receptor that we talked about before? LDL receptor, yes. So LDL receptor, right, is one of the examples for this, protein that can be degraded by lysosome. So LDL receptor that are located on the cell surface that can be internalized, right, into the cell, into the cells, and then protein can be degraded by lysosome inside the cell. And the third category of the protein that can also be degraded by lysosome is a protein that present inside of cell. Intracellular protein can also be degraded by lysosome, right? And lastly, in addition to all these proteins, extracellular cell surface protein, inside intracellular proteins, and the organelle, the old organelle inside the cell can also be degraded by the lysosome. For example, the mitochondria, right? Mitochondria, when it's get damaged and old, and this mitochondria can be degraded by lysosome. And first of all, the damaged mitochondria being engulfed by so-called autophagosome, right? And then fused with the lysosome, and called autophagy lysosome, and then lysosomal enzyme that can degrade all the mitochondria, the organelle inside of this lysosome, right? We have a more detailed the next slide, right? The following slide. Okay. So now let's talk about the first way that in this category, in this lysosome pathway, is vesicle fusion. Extracellular proteins or protein in the plasma, or sometimes the whole microorganism like bacteria, right? Bacteria can be also engulfed or internalized to the, for example, macrophages, right? And whole bacteria can be degraded by lysosome in the macrophage. So this sort of the same mechanism. So either protein in the plasma can be internalized to the different type of cells, or like bacteria can be internalized to the macrophages and be degraded. And the membrane receptors, right, can also be internalized, through the vesicle fusion mechanism into the cells and degraded in the lysosome. Okay, so macroautophagy the pathway in the lysosome polylysis. And macroautophagy pathway usually is responsible for degradation organelles, just I mentioned, right? All the mitochondria, damaged mitochondria. So first of all, this damaged mitochondria, first of all, is surrounded by intracellular membrane proteins to form a so-called an autophagic vacuole, right? Or a different name people will give, like autophagosome, right? So autophagosome basically is a membrane that is surrounded the damaged organelles. And this vacuole fused with the lysosome and forming the autolysosome, right? And eventually the content inside the autolysosome can be degraded by lysosome digest enzymes, right? And this pathway is important in the first stage of nutrient deprivation. For example, the starvation, right? So the cell need energy, and although we cannot get the energy from the diet, right? So the cell need energy. So under these conditions, the cell usually digests this, the self proteins and to provide energy. So under the starvation condition, this process, autophagic lysosome pathway can be very active, right? Just to try to, get the energy for survival. So this is the autophagy lysosome pathway. I give you kind of a idea. So, for example, this membrane can enclose the either the damaged proteins, intracellular protein, and also damage the organelles, for example, mitochondria, right? And eventually form the autophagosome. Right? Autophagosome. So autophagosome basically the membrane structures that enclose the all the damaged proteins or unnecessary proteins or all the damaged organelle mitochondria. And then this autophagosome fused with the lysosome, and we know lysosome has lots of digest enzyme, right? More than 60. So eventually this is the autolysosome, the autophagosome and lysosome fuse together, and so the enzyme that in the lysosome eventually can digest the contents in the autolysosome, right? So this is a process that is very important to degrade damaged mitochondria, to remove all the unnecessary proteins or organelles, right? Yes?
I was wondering, once that last step occurs, are there waste products, or does the cell completely take care of it? Is there waste that...
Waste?
Waste.
Oh, waste?
Yeah. I was just wondering, so that it consumes...
Oh, okay, I see.
And uses it for energy.
Right. Yeah.
Or are you getting...
So, for example, the protein, once it's digested through this process, right? And then release the amino acids. So these amino acids can be used for energy in the cells, or can be maybe synthesize some important necessary protein, essential protein, something like this. So, yeah, good question actually. Yeah. Any other questions? Okay. Yeah, I think here, probably will better indicate this process. And it's just another diagram, a little bit more, more comprehensive to include 4 different ways, right, in this lysosome degradation pathways. So first, like vesicle fusion pathway, I mentioned that can be some, bacteria, right, and be internalized and through the fusion mechanism, or kind of extracellular protein, right, in the plasma can also internalize into cells and form kind of vesicles, right? And these vesicles eventually be fused with the lysosome, and the content in these vesicles could be digested, right, in the lysosome. And the other mechanism, like a macro-autophagy mechanism, right, like damaged mitochondria, can be, enclosed by the membrane and form the autophagosome. Autophagosome fused with the lysosome, right, and damaged mitochondria can be again.
What is the difference between phagocytosis and endocytosis?
What's that? Sorry.
What is the difference between phagocytosis and endocytosis?
Oh, endocytosis means that something that outside the cell, right? And autophagosome, the mitochondria is already inside.
Phagocytosis. The one that talk?
Phagocytosis?
Yes.
Oh, okay. So phagocytosis is basically this process occurs in the mitochondria and specifically internalize the bacteria, right? So this process called phagocytosis. And endocytosis is, the protein, that extracellular protein that internalize into the cell. So actually the process is similar, but it depends on the substrate. Substrate different and give, different name, right? Okay, so and then just like you asked, protein once it's degraded, and amino acids can be used for energy, and also be used for synthesis of essential amino acids, right, in the cells during like a starvation process, right? So this process is very active. Can use your own proteins for survival. Okay, let's take a 10 minutes break.
Are you like BFFs? Why do you love him?
He got so skinny. I didn't even know.
What, was it from the, was he depressed or what? Like why? How do you like me now? I'm on my way. Standing here today.
You want me to finish the water?
Can I drink the rest of that? Maybe something.
Let me smell
Gracias, señorita.
like, I don't want to go find all. Let's try another round. I know I just feel like I would want to be about.
I'll be there.
What's that? I need that. I feel like Matt has that. Is your wife? Yeah. My husband is like very yeah. Oh yeah, totally. Yeah. But I like the opposite.
You like mother, don't you?
Yeah, I like the
Yes, but did you roast them?
yeah. I might have overdone it. They taste pretty good. Oh, they're so good. No, right? Which one's yeah, like that makes it stick without making it mush.
And then roasty. Oh my god. tastes like a sunflower seed.
You think so? No, I don't want to salt on it. Like I want salt on it, but I want to eat some turkey. All that. How much do that?
I'm addicted to these.
Yeah, I just want like just the raw and put them on a pan and put them and then like shake them maybe like.
How long do they have to go in for?
I think I did like 400.
When you put some oil on it, it'll be just fine. That flavor really amplifies like a peanut flavor and stuff like that. It's way nuttier.
Maybe that's only in the spring. Because I tell you, I don't really get any from the fall. But maybe I just never looked at them. I just like believe them. I like doing that mostly because you get candy. Not even because you get candy.
What was the last? Oh, last time was
Oh yeah, was it caramel corn? Yeah, that was good. They all taste, it's so funny, looks like they all taste the same. And then there was the pizza bread.
Oh yeah, you just
Rice probably we didn't
Do meat.
Oh, I don't like that one.
I know. I
With the rice?
Yeah,
Yeah, I hate raw meat. Like I never cook meat. I hate it.
right.
Meat
It's
chips.
nasty,
Well, I
though,
don't
you
eat
think about
like
it.
when it's fine. It's fine, but I hate watching it. Like when I'm out. My dad, he used like kebabs in the summer. And I don't know why he did it. Because like, he put up all the stuff there and everything. Yep. And I was like, Dad, I have to eat chicken. He's like, yeah. And I'm like, and he's like, $20. I'm like, yeah. And it took me like an hour. I was so freaked out. I literally wore this like rubber glove. I put like layers of paper towels on the cardboard like table. Cardboard and I put
so the first one is
we had the bowl, and I like was so gracious to like everything, the salad, and the chicken, and then it was like zero. It took like forever. I'd say it was away. Plus I'm lazy, like I like frozen veggies. We eat like even cutting lettuce is like a big deal. I always let it go back. I never just cut They're new carrots. Ian does it. You want some? They're actually good.
so yeah, because I didn't see
You're the one who took the picture.
Why are you so bitter? Why are you bitter? You are bitter.
I know you are. Just a minute.
Dorothy, you want to do that in class? Yeah. You do?
Oh, I love breakfast together. Just kidding. Nicer. What do you mean? I ran the last 2 days. It's been like 30 degrees. I've been running 40 But I'm actually me and Mario are going to around 2:40. Because we have Betsy at 1:30, so I'm in the hotel. But we have lunch at like 10. Lunch is done at 10:25, so we probably will have to come in by then. We'll be at the bracket.
Yeah, I want to go in and I want to
Okay, let's move on. Okay, I think we finished the 2 mechanisms, right, in the lysosome degradation pathway, right? The fusion, vesicle fusion, and macroautophagy, and then the third mechanism that in this pathway, lysosome degradation pathway, is a microautophagy. That means that the intracellular proteins can also be degraded by autophagosome, right? Autophagic pathways, and so this should be. I think it's a relatively simple compared to the macroautophagy, right? I mean, the intracellular protein that can be, internalized to the lysosome, right? The autophagosome, and then autophagosome fused with the lysosome, and protein degraded by lysosomal enzymes, right? Okay, so the last mechanism in this pathway, lysosome degradation pathway, is so-called direct transport via chaperones, right? Direct transport. So there are several things that's important. First is sequence, peptide sequence. Is very important. That can be recognized by this degradation system. The protein called HSC73, right? It's a specific protein that can recognize the peptide sequence that are located on the proteins, right? So then the binding of this cytosolic tail of the IGP96 receptor. So IGP96 receptor is located on the surface of a lysosome, right? So this receptor can recognize the HSC73 proteins, right? So in other words, the HSC73 protein can bind the IGP96 receptor on the lysosome surface. So eventually that the substrate protein that can be uptake by the lysosome, right? And then protein degraded in the lysosome. And this mechanism is responsible for degradation of 30% of cytosolic proteins, right? For example, the glycolytic enzymes, the proteasome and transcription factor, and this protein in the cells can be degraded by this mechanism, right? Again, 30% of the cytosolic protein is that degraded by this direct transport mechanism. And this slide shows you I think the detailed, probably much better for understanding this process. For example, this represents the pentapeptide sequence, and this is the amino, the protein, right? The protein amino acid sequence, C terminals and N terminals. So this pentapeptides located at the N terminal of this protein, right? And this pentapeptide can be recognized by this protein HSC73, right? So HSC73 is a heat-shock cognate protein, right? This protein has a 73 molecular weight is 73 kilodalton. It's a molecular chaperone, right? It serves that molecular chaperone, can localize in different cellular compartment, located on the that can recognize again this specific peptides, right, on the specific proteins. So again, only the protein that has this specific sequence that can be recognized by HSC73 and can be degraded by this mechanism, right? So the first step, the HSC73 protein recognizes a specific sequence, right, on the specific proteins. And second step is that the HSC73 protein can direct this specific protein to the lysosome, right? So on the lysosome, there is a receptor called IGP96. So this receptor is very important that HSC73 can bind IGP96 receptor on the lysosome surface. Then eventually the whole thing, HSC73 and the whole protein internalize and degrades in the lysosome, right? So this is the mechanism, right? Yeah.
What's the heat shock cognate?
Heat shock cognate. That's a good question. Sometimes a protein is named by, for example, this protein, the expression level somehow can be induced or can be stimulated by heat shock, the process or something like this. So that, and probably at the beginning, the specific function of this protein was not known, but just because the increased expression in response to heat shock, something like that. So that gave the name heat shock protein, something like this. So, yes.
So what is being degraded here? Is the sequence being degraded, or is HSC73, like what's being broken down once it gets into the lysosome?
I think that all proteins, including the sequence, including that specific protein, and also HSC73 cannot be degraded. Yes.
So the sequence is just a specific part of the protein that's being recognized?
Right. Yes. You have additional question? No?
Just any kind of protein?
Yeah, as long as protein has this specific sequence that can be recognized by HSC73 protein.
This is how it'll be broken down.
Right. And then it binds receptor, right? And then the whole thing, internalized, and all proteins be degraded. Right? Yes.
Before step number 1, would you have the squiggly line is the sequence, right?
Yeah.
And then is HSC73, does that come along and bind to it, or is that already
Right.
part of
The binds sequence, that specific sequence, yes.
So like a pre-step would be the squiggly line.
Right.
And then that HSC73 comes along, binds it, and then
Right.
the squiggly line. Okay.
Right. sometimes the mechanism is kind of complicated, you know. Maybe lots of protein has this specific sequence, but the probably buried inside, you know, the protein, then it's not exposed, so will not be recognized by this HSC proteins. But once under some certain conditions, you know, some protein like, you know, unfolded or something, then expose this specific sequence. So then this protein can be recognized, you know. And not like a certain protein has specific sequence, and maybe lots of protein has a sequence, but not always, you know, degrades. All right, so any other questions?
Yeah.
Oh, yeah.
Is HSC an actual protein, or is it just a chaperone?
It's an actual protein. It serves as a chaperone. Yeah. Because it can bind this receptor, right? So it can directly, you know, the specific protein to the lysosome surface and bind the receptor, and then hosting can be internalized. Okay, so this sort of kind of just summary, I guess. So the lysosome pathway, right, can degrade extracellular and cell surface protein, and also intracellular proteins and organelles, right? And there are 4 different ways in this particular pathway that, you know, protein can be degraded. First, the vesicle fusion and macroautophagy. So basically, macroautophagy responsible for degradation of organelle, right? And microautophagy degrades intracellular proteins, and chevron-mediated autophagy pathway is, you know, the degraded cytosolic protein that has specific, you know, that sequence, right? Okay, now let's move on to the second main pathways of protein degradation, which is proteasome pathway, right, and degrades proteins from the cytoplasm, nucleus, and ER in the cells, right? So proteasome pathways is responsible for 80 to 90% of intracellular protein degradation. So again, the 80 to 90% of intracellular proteins are degraded by proteasome pathway, right? So proteasome, here is an image you can see. Proteasome has a 2 parts, right? Core particles and the regulatory particles. So core particles and regulatory particles. So in the core particles, actually 2 sets of 14 proteins. So you can see that there are actually 4 rings, right? Each ring contains the 7 enzymes, and the 2 rings they are identical. I mean, you know, alpha and beta rings, and the other alpha beta rings, they are actually identical, right? It's 2 rings. And then in the regulatory particles, which in the you know, 2 ends, each regulatory particles contain the 6 ATPase, right? And it's important, you know, for allergies. And these regulatory particles also contain the recognition of specific protein. They call it ubiquitin. Ubiquitin is a protein that localizes in the regulatory particle, and the ubiquitin is very important protein that can recognize specific protein, right, and direct this specific protein for degradation by proteasome. So again, the proteasome degradation is responsible for the regulated degradation of important proteins that controls cell growth, proliferation, cell differentiation, apoptosis, and immune response, right? So the protein in the cells that regulates the important functions that usually need to be, again, the turnover, right? So degradation of these proteins is also the part of a regulatory mechanism. Is that clear? I mean, because these proteins that control, cell growth, proliferations, differentiation, and immune function, and the concentration of these proteins very critical, right? We don't want to have too much or too high levels of this protein, so the degradation is part of the mechanism to control the levels of these critical proteins, right, or factors. And so again, the proteasome is a very important pathway that regulates the levels of these critical proteins, or regulatory proteins, right? So of course, because of this protein that involve, important functions, so the proteasomal degradation pathway can control cell cycles, right, and can also control the immunogenic peptides, right, and also concentration of the enzymes and transcription factors, right, in the cells. So what signals in protein that can be recognized by the proteasome and initiate degradation? So that's a question. I mean, proteasome degrade protein, but not all type of protein, right? Otherwise, it will be messed up. So the protein that I just mentioned, right, the main mechanism is the ubiquitin, right? So the proteins need to be attached to ubiquitin proteins. So ubiquitin protein, again, is a very important protein that once this cell decided to degrade that protein, that ubiquitin will attach to that specific protein and direct that protein to degradation by proteasome. So again, ubiquitin is the main mechanism in the proteasome degradation pathway. Yes?
Does the ubiquitin protein just only recognize that list of that list on the previous slide about functions of proteasome?
Yeah.
That what only it recognizes and nothing else?
Right, right, yes. And in addition to ubiquitin mechanism, some proteins, if have a specific amino acids motif, that can also be degraded by, recognized and degraded by proteasome, right? And the other mechanism is that the altered structure, for example, some protein undergo the oxidative modification or expose of hydrophobic amino acids, the structure modified protein, right? As I just described before, some specific hydrophobic amino acids exposed, protein unfold, and specific hydrophobic protein exposed, amino acid exposed, and this type of protein can also be recognized by proteasome, right, and degraded. So again, under the certain conditions that when protein structures change, and this protein can be recognized and by proteasome and degraded. Now let's talk about ubiquitin. So ubiquitin is a small protein, right? Highly conserved, and almost it can present in the all type of cells, right? So it's universally expressed or present in all type of cells, and it present in the cytosol of the cells, ubiquitin, and it's used to target proteins for degradation, right? So again, the main function of ubiquitin is to target specific protein for degradation. And so ubiquitin-dependent degradation is involved in the degradation of proteins that are so unassembled. The protein, once synthesized, if for some reason, made a mistake that this protein are unassembled, and this protein need to be, removed, and this type of protein is also undergo, ubiquitin-dependent degradation. And the second type of protein, like damaged or misfolded proteins, right, is also being targeted by ubiquitin and for degradation. And the third category of protein, that the protein that have a short half-lives, need to be degraded frequently, and this type of protein can also be recognized by ubiquitin and degraded. So first, the regulation of proteasome activity. So how proteasome activity is regulated? Again, we don't want to proteasome activity like, constantly active, right? Otherwise, all the protein will be degraded. So only under certain conditions, right? We need a proteasome activity to degrade proteins. So first is phosphorylation of a certain domains in the target protein. So once some target protein gets phosphorylated, right, it can be recognized by ubiquitin and degraded. And glycosylation basically inhibits degradation, to protect proteins from degradation, right? So again, the phosphorylation usually stimulates degradation, and glycosylation inhibits degradation, right? So this also is a regulating mechanism to protect, protein from the degradation by proteasome, phosphorylation or glycosylation, right? And the proteasome activity can also be regulated by hormones, like insulin basically inhibits proteasome activity, so inhibits protein degradation, right? And glucagon, cortisol usually stimulates the proteasome activity, so right, can degrade, promote the degradation protein by proteasome. And also the disease, injury conditions that proteasome activity can also be activated, right? So you have more proteins being degraded during the disease, right? If you're losing weight and so again, the proteasome activities is regulatory, right? It's regulated by either the protein itself, modification, phosphorylation, glycosylation, or can be regulated by the hormone levels, right? Depend on fat fasting condition, hormone levels, and also disease, injury, condition can also regulate proteasome activity. Okay, so this is, I guess, the last pathway, main pathway for protein degradation, right? The cytosolic proteinase. So in the cytosol, inside of cells, we actually have many proteinases that can also degrade proteins, like cellular proteins. Just give one example here, the kelpins. So kelpins is the proteinase, right? Calcium-dependent proteinases. So calpains partially degrade membrane and the cytoskeletal proteins, right? And also several membrane-associated enzymes. So basically, degrade membrane protein and cytoskeletal proteins, and also some enzyme that associate with the membranes, right? And this, in general, some membrane-associated proteins, right, can be degraded by the calpains. The calpain activities, again, is not always active, right? It's regulated. So regulated by calcium and inhibited by the other protein called calpastidine. Calpastidine is the protein that can inhibit calpain's activities, right? Regulate calpain activities. And this pathways is also pretty active in the muscle and basically responsible for the degradation of myosin. This diagram just tells us the process, how calpain degrades proteins and how this process is regulated by calpastidine. As I mentioned, calpastidine is the protein that has an inhibitor function, right? It inhibits calpain activity. So calpain is again is a proteinase, right? Directly degrade proteins, and under certain conditions, the calpain bind to the calpastidine, and once it's bind to the calpastidine, and the activity is inhibited, right? And calcium is a factor that can trigger or stimulate the calpain activity. So when we have a higher calcium level or calcium concentration is increased in the cells, so can promote association of calpain with the calpastidine. So again, the calcium, if we have a higher levels of calcium in the cells, that will cause the release of the calpain from calpastidine, right? So then the calpain become active, so then can bind the target proteins that degrade the proteins. So again, the calpain activity is regulated by two things. One activates and the other one inhibits, right? The calcium stimulates calpain activity, and calpastidine inhibits calpain activities. Yes?
Do you say that calpastidine releases the calpain in response to high calcium?
Yes.
To release?
Right,
Okay.
right.
But
Yeah.
it also inhibits it?
Well, once it's not bind together, then the inhibition effect will be gone. So then make it active.
Right. Okay.
Yeah. Okay. Any other questions? So this pathway seems kind of relatively simple, right? It's two things regulates it. Okay, so pretty much all things that I have for today, but just go through the summary. So summary, we talk about, I think that we talk about the protein turnover, right? Protein, maybe I can go back to the very beginning. So we talk about the protein, the metabolism, the amino acid pool in our body, right? And amino acids can be metabolized through the several different pathways. The one is protein turnover. So amino acids can be used for synthesis protein. All protein can be degraded to release amino acids, right, and contributing to the amino acid pool depend on, again, the physiological conditions. So, fat condition, you have a little bit you have a high activity of protein synthesis, and fasting condition, you have a high activity of protein degradation. So protein turnover, between amino acids pool and protein. And amino acids can also be metabolized, further metabolized, right? And again, also depend on the physiological condition. Under the fat condition, that amino acid has actually additional element like nitrogen. So nitrogen, it's different from the glucose fatty acids carbon metabolism. So first of all, the nitrogen need to be removed and be metabolized through the formation of ammonia and urea synthesis. So Carrie is going to talk about more about details the nitrogen metabolism, right? Amino acids. And in terms of the carbon metabolism, again, depend on the physiological condition. Fat condition, excessive amino acids can be metabolized in actually 3 different ways, right? Pathways. One is just complete oxidized, to acetyl-CoA, enter TC cycle for ATP production. And the second pathway that the amino acids metabolize to acetyl-CoA and enter TC cycles and use the TC cycles for synthesize oxaloacetate and then go to the gluconeogenesis for glucose, for synthesis of glucose, and glucose eventually being converted to glycogen in store, as a stored energy, right, in the fat condition. But of course, under the fasting condition, this glucose will be released into the circulation, right, contributing, energy for the other tissue use. Yes.
So are these 3 pathways then there in the fasting condition?
No, this will be different. I'm sorry, I probably need to go to that slide. I'm just talking about the very beginning, the slide, the whole about how amino acids or proteins metabolize in the body, right? If we still remember. And then and also, the protein amino acids can be oxidized to acetyl-CoA, and acetyl-CoA being used for fatty acid synthesis, triglyceride synthesis under the fat condition, right? So that was protein or amino acids metabolism. And then we also talk about the protein turnover, right? Synthesis and degradation. So synthesis is just relatively brief. I think we just mentioned that the protein can be synthesized through, and we talk about actually the transcription, translation process, right? And how, what kind of component or factors that involved in the protein translation. And then I think we focus on more, we talk about more about the degradation, right? Protein degradation process. So this are we mentioned the 3 main pathways of the protein degradation, right? Lysosome pathway, proteasome pathway, and cytosolic proteinase pathway, right? And lysosome pathway that has 4 different mechanisms, right? The vesicle fusion mechanism, and macro autophagy mechanism and micro autophagy mechanism, and then the direct transport via chaperone mechanism, right? And each mechanism that degrades a different type of proteins, right? And in terms of a proteasome pathway, then we said the proteasome pathway is responsible for degraded 80 to 90% of intracellular proteins. That protein can be in the cytosol, can be in the nuclear and ER. And the main mechanism for proteasome degradation that involves the ubiquitin protein, right? So ubiquitin basically targets the specific protein and direct that protein for the degradation. I think that's all that I have for today. Do you guys have any questions? Yes. Yeah. Well, I mean, I think Carrie will send me the notes and also study guides. Yeah, we'll post hopefully today or maybe tomorrow. Sorry?
Is this the only presentation, or are there going to be any others?
I mean, for amino acid metabolism? Sorry? Yeah, 3 lectures, and then we also have an exam for protein amino acid metabolism.
This is the only presentation that is out of Moodle. Are more to come?
Sorry, I probably didn't quite understand.
The only presentation that I saw at Moodle.
Yes, right. I mean, the amino acids notes have not been posted yet, but will be posted.
Okay.
Yeah. Yeah.
Quick question.
Yeah, sure.
For this one, I just want to clarify to make sure I'm not knowing this wrong. It's asking what is the donor of the 2-carbon unit for fatty acid synthesis? And I know that the first 4 carbons usually in a fatty acid come from acetyl-CoA, but then the donor of the 2-carbon units is the malonyl-CoA from there on out.
Yeah, right.
And that got marked wrong.
Oh, I think you should get the 1 point back.
Yeah.
I
I can just leave that with him and that's fine, yeah?
Yeah, okay.
Perfect. Okay,
Yeah.
thank you.
I think you probably also have to correct.
Oh, okay.
Hi, I'm Sarah. I'm supposed to
Sarah.
take the exam.
Oh, yeah, okay, sure. I think that we have yeah, just could you wait for me?
Yeah, where do you want me to go?
1:15. Yeah, maybe you can do it.
1:15?
Yeah.
Okay, thank you.
Yeah.
I was just wondering when the first homework assignment will be graded.
Oh, Derek, could you answer the question?
Can I ask a question about
Yeah.
the quiz? So I thought malonyl-CoA was the donor for.
Okay, so you should get 1 point back. Can
Oh,
you
okay.
give it to?
Yeah, for sure. Thank you.
Yeah. Hi.
I wonder if I can come look at my exam.
Sure. Yeah.